Okay, very good morning, my dear friends. This is Dr. Padala Tirupati. In the regarding of CPGET 2024. So, my dear friends, very short period is here. Don't waste your valuable time. So, today our eminent resource person, Dr. A.KAVITHA Madam Garu, Assistant Professor, Department of Botany, RBVRR Women's College, Autonomous, Hyderabad. Madam is going to explain about the Pteridophyta type study. Yesterday, Madam has clearly explained about the ecosystem, morphology, anatomy, reproduction, life cycle. And also, today, Madam is going to continue today Marselia. I hope Marselia. Am I right, Madam?
Yes, sir. Yes, sir.
The type study Marselia, it is also most important sporocarpus structure and morphology, anatomy, reproduction, life cycle. So, Madam, please welcome and share your screen. Start your session, Madam. Please welcome.
Okay, sir. Thank you, sir. Okay, good afternoon, my dear friends. So, today we are going to continue with another type study of Pteridophyta. So, that we call it as the Marselia, which is a very important topic for the entrance exam point of view. Just I'm start sharing my screen. Okay, let's start sharing my slideshow. Yeah, I hope you can see the slide over here. Anybody please respond? Can you see the slide?
Yes, Madam. It's clearly visible. Please proceed, Madam.
Yes, sir. Yeah, this is a Marselia. We are going to talk today about the type study. So very commonly we call it as this is called as the water fern. Like that means to say that this is a pteridophytic plant which belongs to the aquatic group. So whereas we talked about yesterday the the equisetum and the lycopodium, which are the terrestrial group, that is the terrestrial pteridophytes growing on the land surface. But whereas this Marselia, it is a hydrophyte. It is growing in the water surface. So Marselia, it is an aquatic or semi-aquatic plant. So it are commonly grown in the temperate regions. So as we all know that where there is a moist conditions where it can grow. And it grows always in the freshwater ponds and the ditches. So the best example of this Marselia species, what we can call it as Marselia quadrifolia. So the species name quadrifolia, it refers to the presence of these 4 leaflets. You can see here, the leaf is getting dissected into 4 leaflets. 1, 2, 3, 4, 4 leaflets. So that is the reason why this Marselia species is named as Marselia quadrifolia. So this Marselia quadrifolia and Munilia minuata, these are commonly found in the Pakistan regions. And when you go for the morphology of this Marselia plant, so the vegetative plant, as we all know, we call it as a sporophytic plant. And this plant, it is differentiated into roots, rhizome, and the leaves. So here, as we say that as it is an aquatic plant, so the stem is getting reduced into the rhizome structure. So that's why we call it as a rhizome. So you can see here, this is a plant body of the Marselia. So this is the, so you can see here the brown color one and here the picture. So the one which is growing horizontally, this part we call it as the rhizome. And the underground part of this rhizome are emerging are the roots. And from this rhizome, we can see the development of the leaves. So this is a petiole and these are the leaflets. Now, so when we discuss about the rhizome here, yes, any doubt over here? Okay, now So the stem here in the Marselia plant, it is in the form of the rhizome. So this is going to have a growth of unlimited growth. That is, it will be growing unlimitedly. And the rhizome here, it is a dichotomously branched and it has nodes and internodes. You can see here the two branches are rising from the same branch. So we call it as a dichotomously branched and it has nodes and internodes. And at the point of each node, we can see the development of adventitious roots towards inner side and the leaf which is going to arise on the dorsal side. So you can see here. So this is a rhizome here. It is divided into nodes and internodes. So this is the point, what we call it as a nodal point or the nodal region. Now, this is the dorsal side. So you can see here, the above one, this is the dorsal side. And the below, we call it as the ventral side. So from each node, from the ventral side, the roots are going to be arising. And on the dorsal side, we can see the development of the leaf. So adventitious root and a single leaf we can see arising from the each node on the dorsal side of the rhizome. So that is the external morphology. Now when we talk about the leaves over here, the leaf structure, and I told you that these leaves are compound leaf. They are not simple. As here the lamina is getting, why are calling it as a compound? Because the leaf has been dissected and it is divided into 4 leaflets. Okay, so you can see here 1, 2, 3, 4. The complete leaf lamina is dissected into 4 leaflets. So that is the reason why we call the leaf as a compound leaf. Now, and each leaf, it is going to have a very long petiole. And these leaflets, what we call it as calyx, or these leaflets, they are arranged in a cross-like manner at the tip of the petiole. So cross, you can see here, they are in the form of a cross-like manner. And each leaflet, it is having a triangular shape. And the veins are showing a reticulate venation. And the stomata are being located on the dorsal side and also on the ventral side of the leaflets. So you can see here, this is a very long petiole. And the tip of the petiole, the lamina is getting dissected into this compound leaf, having the presence of 4 leaflets. And each leaflet here, it is showing a triangular in shape, very clearly the triangular shape. And each leaflet is going to have a single midvein and the lateral veins coming from that. So we call that as a reticulate venation. And these leaflets are arranged in the form of a cross. So this is the external morphology of the Marselia plant of the roots, adventitious roots, the rhizome, and these are the leaves and the leaflet structure. This is the external morphology. Now, when we go for the internal morphology of the rhizome and as well as the leaf, so when we talk about the internal structure of the rhizome, it is going to composed of epidermis, the outer layer, the cortex, as well as the central stele. Now, when we talk about the cortex here, a very important point to be remembered here in the cortex of this, so this is a cortical region. Okay, this is the region which is lying below the epidermal region. So that we call it as a cortex. So you can see here in the cortex, there are so many air spaces. Okay, there are gaps here. In between them, we can see the parenchymatous, a longitudinal parenchymatous tissue. So the air spaces which are present here in the cortical region, it constitute the parenchymatous tissue. So why this presence of these air cavities or why this portion, why we calling it as the parenchyma? Because it is going to store here this parenchymatous tissue is going to store the air in them. Why? Because this Marselia is an aquatic plant. It has to float in the water surface or on the water surface or inside the water. It has to grow or float. In order to float, the plant has to develop an empty space. It should be light enough, just like a balloon. Just like a balloon, how a filled balloon, the balloon which is filled with air, how it will be, it will sink in water or it will float on water. So due to buoyancy, it will float on the water because it is completely filled with air. So it becomes lighter in weight and it floats on the water. So the aerenchymatous tissue plays a role of buoyancy here. It will make the plant to float on the water surface. So the rhizome of the Marsilia plant, the inner anatomy, that is the cortical region of the rhizome, is going to have the presence of aerenchymatous tissue. Due to the presence of that, the plant will float on the surface of the water. So this, you can make a note of these, girls. Very, very important here. The presence of aerenchymatous tissue in the rhizome is an hydrophytic character. So this is a question. What is the hydrophytic character of the Marselia pteridophyte? So presence of the aerenchymatous tissue in the rhizome, which helps the plant to float on the surface of the water. Next, the inner portion of the cortex here, it is going to compose of sclerenchymatous cells, providing the mechanical strength. And the inner side of the cortex, we can see the presence of a single layer endodermis. And inner to the cortex, we can see a stela region. And here the stele is defined as a solenostelic condition, amphifloic. Yes, ma'am.
Ma'am, can we write these points, ma'am?
Yeah. This point you can note down. The hydrophytic character of Marsilia is, so I told you that Marsilia is a hydrophyte, right? So it
Yes,
is a
ma'am.
aquatic plant. Okay. So what
Okay,
is,
ma'am.
why I am calling it as an hydrophyte? By
Because ma'am, I'll tell you, ma'am. It
yeah,
lives in water.
yeah, it lives in water. And how the plant is floating? Is it? It will be floating on the water. How the plant is floating on the water? Because it consists of air chambers or a tissue, what we call it as aerenchymatous tissue. So with
Yes,
this
ma'am.
tissue, it is able to float on the surface of the water. Okay.
Ma'am,
Hmm.
ma'am, how plant can float in water, ma'am?
That is the reason why I'm telling now here. In the rhizome, in the internal structure, okay, inside
ma'am.
the rhizome, when I take a transfer section, okay,
Okay, ma'am.
so inside the rhizome, you can see the empty spaces here. These are the air chambers. You can see here the empty spaces. You can see this arrow, the pointer.
Yes, ma'am. Yes, ma'am.
Yeah. So these are all are the empty chambers, what we call it as. Or it is also called as aerenchyma. Okay. So you see here, this is the aerenchyma, empty spaces. So due to these empty spaces here, the plant body can float very freely in the water surface as it will filled with air. Got it?
Yes, ma'am, I got it.
Yeah. So, and another point here, the stele of Marsilia. You can take down this point also. The stele of Marsilia is regarded as amphifloic solenostele. Now, what is this solenostele here? Presence of a central pith region in the center and having the different layers of xylem and phloem. So you can see here, the xylem and phloem, it is divided into inner xylem and outer xylem, and inner phloem and outer phloem. Okay, the vascular tissue is divided into two layers or two vascular bundles with a central pith region. So this condition is called as amphifloic solenostelic condition. So this is also an important point to be noted. The stele of Marsilia, whereas the stele of Lycopodium as well as the stele of Equisetum, it is a protostelic condition. We can't see any pith in the center. Okay, so that condition is called as protostelic condition. But here, Marsilia, it is a solenostele having a central pith region, and that too it is of amphifloic solenostele. Presence of inner phloem and the outer phloem with a xylem in the center. I hope this point is clear. Right. Next, moving on to the petiole, the transverse section of the petiole. So this is the petiole diagram here, you can see. So even the petiole also, the having an outer cortex, outer epidermis for protection. And we can see the cortex region here. The outer, we call it as a outer cortex here. This outer cortex is a single layer. And below this outer cortex, we can see the presence of aerenchymatous tissue, which is also filled with air. And below that, we can see the presence of the inner cortex, parenchymatous, inner cortical region. And in the center, you can see the presence of the stele. And there are 2 points here, girls. Remember, Marselia rhizome, the stele of Marselia rhizome is of solenostele condition. I'm repeating twice here, girls. The rhizome, in the rhizome, the stele is amphifloic solenostele, whereas the petiole, the stellar region of petiole, it is of protostelic condition. So you can look into this here. Protostelic condition. That is, there is no pith in the center. No, there is no pith region. Am I clear? There is no pith region. It is completely the phloem, and along with the phloem, we can see the presence of the xylem. Okay, 2 distinct arms. That is, we can see the presence of 2 xylem vessels, which are in the form of a V-shaped. So you can see here, this is the xylem elements here, which is in the form of a V shape with a central metaxylem and protoxylem towards the arms. That the tips of this metaxylem, we can see the presence of the protoxylem. And you can see here there are 2 xylem elements, and in the center completely filled with the phloem. So there is no pith in the Marselia petiole. That is the reason why we call the stele of Marselia petiole as a protostele condition. This is clear, all of you? Right. So this is the transverse section of the Marselia petiole. Now, when I go for the transverse section of the leaf or the leaflet, when I talk about, so the leaflet, it is showing a dorsiventral leaf-like structure. This is the dorsal side and this is the ventral side. So we can see an upper epidermis and the lower epidermis. And below the upper epidermis, you can see the palisade tissue. That is, the cells are elongated in shape with chlorophyll tissue, with the chlorophyll pigment and presence of chloroplast in these palisade tissue. That is the reason why the dorsal surface of the leaf is greener in color. It is green. And you can see the ventral side of the leaflet, what we call it as a mesophyll tissue. And this mesophyll tissue here is going to have the presence of air spaces again. Okay, so that is the reason here this is also called as parenchyma. And here the upper epidermis and also the lower epidermis, we can see the presence of stomata for the process of exchange of gases. And in the center, we can see a single vascular bundle here in the center. And the epidermal region is going to surrounded by the cuticle. That is again one more hydrophytic character. So the complete plant, that is Marselia rhizome, Marselia petiole, and also the Marselia leaflet. In all these parts, in the anatomy of all these parts, we can see the presence of aerenchymatous tissue. So that is the reason why the plant can able to float in the water, and that's why it becomes a hydrophyte. Okay, anatomically, presence of this erenchymatous tissue makes the plant to float freely on the surface of the water, and hence it becomes a hydrophyte. Next. So that is about the external morphology of the Marselia plant, and also about the internal morphology, that is the anatomy of rhizome, anatomy of the petiole, and anatomy of the leaflet. Next, we are moving on to the very important point of the Marselia, that is reproduction. And that we are going to discuss about a very important structure called as a sporocarp. Now, just I'm showing you here the diagram of the Marselia again. So here, at the base of the petiole, or at the nodal region of the rhizome, at the time of reproduction, we can see some seed-like structures. Okay, some brown kidney-shaped structures or brown-colored structures developing at the base of the petiole or at the point of the nodal region of the Marselia plant. Those structures are called as sporocarp, or this is also called as the fruiting body. I repeat this once again. At the time of reproduction, at the base of the petiole, or at the base, or just the point near to the nodal region, we can see the presence or the development of some seed-like structures or bean-shaped structures. Those are called as, we call them as fruiting bodies. Those are called as sporocarp. Now, what is the sporocarp here? This is a fruiting body which is going to consist, or which is inside the sporocarp, we can see the presence of sporangia. Now, these sporangia, they develop into spores. Okay, now, so the sporangia which are developing in this sporocarp, we can see the development there. We can see the presence of two different types of sporangia inside the sporocarp. Those are called as megasporangia and microsporangia. So that is the reason why the Marselia plant is regarded as heterosporous. That is why we are calling it as a heterosporous plant, because on the spore, that is within the sporangia, or the Marselia plant is going to develop two different type of spores. Those spores are called as microspores and megaspores. And these microspores and megaspores, they are developed inside respective microsporangia and megasporangia. These sporangias, where they are, these microsporangia and megasporangia, they are present inside the sporocarp. Okay, and how is the sporocarp? It is a bean-shaped structure, and it is a very hard and having a stony wall. So just I'll show you here the diagram. So you can look into this. This is the diagram of a sporocarp. Okay, and these are, so you can see here, these are all the locule, or we can call it as a chamber inside the sporocarp. All these are the sori. We call it as the the sporangia. We call them as sori. And inside the sori, you can see the presence of sporangia. This is a megasporangia, that is a bigger one. We call it as the megasporangia. And the smaller one, we call it as the microsporangia. I hope this is clear. So both the sporangia, megasporangia and the microsporangia, they are developed within the same sori or within the same compartment of the sporocarp. So that is the reason why Marselia is called as a heterosporous plant. Now, the spora, so you can see here, the sporocarp, it is divided into two inner chambers, and in each chamber, we have a row of sori, and each sori of these, they alternate with each other. And the sori will develop a row of megasporangia and microsporangia. So this is a diagram I will show you. This is one compartment, one chamber, and this is another chamber. And in each chamber, we can see all these are the sori. And in each sori, you can see the presence of megasporangia and the microsporangia. So this is again a beautiful diagram of the sporocarp. Okay. So if I divide the sporocarp into two halves, this becomes the topmost one becomes the first chamber, and this one becomes the second chamber. And in each chamber, you can see one sori, okay, alternating with each other. This is one sori. And in each sori, you can see the development of megasporangium and the microsporangium. Right. Okay, so the megasporangium here, it contains only a single megaspore, whereas microsporangium contains, or it will develop 32 to 64 microspores. Okay, there are so many microspores which are developed inside the microsporangia, whereas the megasporangia, it develops only a single spore, that too we call it as a megaspore. Now, so how this sporocarp is developed? It is believed that sporocarp is having a single folded pinna. That is, this pinna has a single vascular bundle. The receptacles or the placentas, they are produced on the ventral side of this pinna, and the receptacles are going to bear these sporangial initials, and each receptacle with the developing sporangia, it forms an outgrowth or towards the midrib side of the pinna, and this covering, it is known as the indusium. Now, how this sporocarp is dehiscing? The mature sporocarp, it will open after 2 to 3 years. The stony wall of this sporocarp, it will decay and it will open the sporocarp, and the inner gelatinous material, it will absorb the water and it will swell. And as a result, the sporocarp will split into two equal half, and the gelatinous cord or the sporophore, it will absorb the water and it will swell. And the spores, due to the splitting of the sporocarp, the spores will enter into the sporocarp, and the spores will germinate. It will be remain viable for a long period of time, and these spores will develop into the respective gametophyte. So the development of sporangia is not that important for you all. Now, so how the male gametophyte is developed here? So as I told you, microsporangia will develop 32 to 64 number of microspores. Now, as the spores, they enter into the water, each microspore, it will divide into two cells. The smaller cell becomes prothalial cell, and it is reduced male gametophyte, and the larger cell will develop into two antheridial initials. Now, each antheridial initial divides to form the androgonial initial, and the androgonial initial of each antheridium, it will divide and forms anthrozoid mother cells, 16 anthrozoid mother cells, and each anthrozoid mother cell will divide and develops anthrozoids, the so-called male gamete. And these anthrozoids have many coils with a single flagella. This is again another important point here. This is the structure of an anthrozoid here. The anthrozoids of Marselia, it is single flagellated, whereas the other cells, they are biflagellated. In Lycopodium and Equisetum, the anthrozoids are biflagellated. But here in Marselia, the anthrozoid, it is a single cell, it is highly coiled with a single flagella. Okay? Now, so these anthrozoids, as it is going to have the flagella, they become free and it will be floating in the surrounding water. Whereas megaspore, when we talk about the development of the female gametophyte, in the megasporangium, we can see the presence of only a single megaspore. And this single megaspore, it divides into two cells, the smaller one and the larger one. The smaller cell, it occupies the complete beak, whereas the larger cell, it does not divide further. This smaller cell will function as an apical cell, and this apical cell will divide and cut off to form the vegetative tissue, and this apical cell remains functional and forms the archegonial initial. And this archegonial initial, it is going to develop into the archegonia. Okay, so it will going to form four neck canal initials and further forms a neck. Central cell will forms a venter canal cell, and the larger primary cell will forms a venter. Now the primary canal cell produces the two neck canal cells, and the venter canal cell divides to produce a large egg cell, a lower egg cell and an upper venter canal cell. Now this egg cell will function as the female gamete. Now the venter canal cell and the neck canal cell, upon maturity, they will disintegrate, forming an opening for the anthrozoids to fuse. Okay, so this is a single megaspore, and the single megaspore will form the archegonial initial and developing. So you can look into this. This is the archegonia which is developed, and this is a central egg cell or the female gamete, and this is the venter canal cell and the neck canal cell. Upon maturity, they will degenerate, and from here the neck, the anthrozoids will move and fuse with the egg. So fertilization here, upon maturity of the megaspore, the several anthrozoids, they enter into the megaspore where we can see a mucilage, and one of these anthrozoids, they enter into the archegonium. It will fertilize and it will fuse with the egg to develop into the oospore, or we call it as a zygote. I hope this is clear for you all. Now, the oospore or the zygote, it divides further, producing 4 cells, among which the 2 cells will develop the stem and the cotyledons. The another 2 cells will develop into the foot and the root region, the so-called the rhizome and the root. Now, so it is 2 to 3 cells, the gametophyte formed, it is 2 to 3 cells in thickness, and this calyptra, it develops into the embryo, and the surface cells of this calyptra will develop along rhizoids, the cotyledon and the root, they grow faster than the calyptra, and the root enters into the soil. The cotyledon expands to form the first simple leaf, and the primary root is going to be replaced by the adventitious root, and the stem grows horizontally on the soil, forming the rhizome. So that is how the zygote and the embryo will develop into the first formed plant of the sporophyte. Now here, alternation of generation we can observe. The sporophyte and the gametophyte generations, they both are getting alternating with each other, and the vegetative plant of the Marselia, which is going to form a diploid sporophyte, and which is a heterosporous type again. Okay, so the microspores here giving rise to a male gametophyte and the megaspore giving rise to a female gametophyte. And both male and female gametophyte, they complete their development within the spore wall. So as a result here, the oospore, whatever is going to develop, the so-called zygote, it develops into the next generation of the sporophyte again. So this is the life cycle of the Marselia very clearly seen. So here the adult Marselia sporophytic plant. So at the time of reproduction, it will going to develop the fruiting body. This is the sporocarp. Now inside the sporocarp, you can see the presence of microsporangia and the megasporangia. Now, so the microsporangia will develop the microspores and the megasporangia will develop the megaspore. Microspore upon germination forms the male gametophyte, and this male gametophyte will produce the antheridia, the anthrozoids. These are the anthrozoids, the male gamete, and the megaspore will develop the female gametophyte here. So within this female gametophyte, we can see the development of archegonia. So within the archegonia, we can see the egg and the venter. So the development of the egg we can see here inside the female gametophyte. So these male anthrozoids, they will enter into this archegonia, fuse with the egg, undergo fertilization, and form this zygote. Now this zygote will develop, upon development, forms the young plant, the young sporophyte. Upon maturity, that is this young sporophyte will develop and forms upon maturity, it develops into form a adult sporophytic plant again. So this is about the life cycle of the Marselia, the reproduction, the alternation of generation, fertilization, reproduction, anatomy, and all. So this is about the type study of the Marselia. So if you have any doubts, please, you can. I'm not stop sharing here. If you have any doubts, please at any point. And just I'm moving the slides over here. Any slide, if you want to be repeat again, I will. So this is the anatomy. I'm moving on from the starting slide onwards again. So this is the external morphology, how the Marselia plant looks like. Now this is the external morphology of the plant. Next, we are moving on to, this is the anatomy of the rhizome. Next, anatomy of the petiole. And this is anatomy of the leaflet. Next, moving on to the reproduction. This is the structure of the sporocarp. And this is a development of the sporocarp here. The sporocarp developing into the gametophytic tissue. Development of male gametophyte here, how the microspore, it is dividing and forming the antherozoids. Female gametophyte, how a megaspore, it is developing into the archegonia or the female gamete. Fertilization, and after the fertilization, the zygote is developing into the young sporophyte. So this is completely here. This generation is sporophytic generation and this generation is a gametophytic generation. So sporophyte here alternating with the gametophytic generation. So diplohaplontic life cycle. So I'm stop sharing here. So any doubts here, please? Am I audible? Yes, audience, please. Any doubts in this Marselia life cycle? Yeah, please respond. Hello, am I audible? Okay, no doubts. Okay, very good. Naina. Sorry, Nafi Sultana. Okay, ma'am. I hope there are no doubts for this. Sir, I have done with my topic here. Hello, sir, I am done with my topic. Right. Okay, then I am ending my session here. Thank you very much, ma'am.
Okay, madam, thank you very much. Regarding of today's class, if you have any doubt, my dear friends, please ask our respected madam. So madam is going to clarify your doubts. Otherwise, I will going to wind up the session. Okay?
There are no doubts, SM sir.
Okay, thank you. Thank you very much, madam. Nice session, wonderful presentation. Today's beautiful slides are shown. Thank you very much, madam. So
Yes.
tomorrow we will meet.
Yeah, thank you, sir.
Thank you, madam. Thank you.